Gel electrophoresis. This is a very important technique which is used in the process of recombinant DNA technology. It is actually the separation and isolation of DNA fragments after they have been acted upon by the restriction enzymes. Now, when a restriction site is being acted upon by the restriction enzyme, the desired pieces of the fragments of DNA are obtained. Now, out of all the DNA fragments which are being obtained, not all are useful. So, it is a technique which is used to separate and isolate the DNA fragments obtained after the use of restriction enzyme. In a way, we can say that it is a method of purification. It is purification of DNA fragments. And this technique was developed by Dacelius. If we try and study, or let us now get into what is actually gel electrophoresis. Now, the DNA we know is a negatively charged molecule, and it is this property of DNA we are going to utilize in this process. Also, the second important factor is the density. So it separates, in this technique there is a separation of the molecules on the basis of their size, that is density gradient separation, and the negatively charged DNA. We are using both these properties in this gel electrophoresis to obtain the fragments of DNA of our requirement. Now, for this we have to use a matrix or a gel. Now this matrix or a gel is placed on that agarose gel. Agarose gel is obtained from the seaweeds. So if we see the arrangement of this, the plate, these are the wells over here. Wells over here, and let us name it as 1, 2, 3 and 4 over here. Then this is the matrix or the gel, that is the matrix over here. This gray portion, or I am making another square, this is the matrix. These are the wells. This is the matrix. Then there are the DNA fragments are placed over here. These are the DNA fragments shown in red. When we are trying to understand, this is towards the high base pairs, that means high density, and these are of low base pair, low density, right? And this is the negative side, this will be the positive side, that is the, we are talking of anode, and this is the gel plate. So over here in this technique, since the DNA molecules are negatively charged molecules, they can be separated by making them move towards the anode. So this end is the anode and this end will be the cathode. They will be moving under the electric field through a medium or the matrix, and the matrix commonly over here is agarose. Agarose is the matrix which is obtained from the seaweeds as told earlier, and the matrix is spread evenly and thinly on the glass plate. So this is the matrix and this is spread over on the glass plate evenly, and now it will be known as a gel plate. The phosphate group actually in the DNA are negatively charged, because we have studied the structure of DNA, and the phosphate group which is present over there is negatively charged. So it will move towards the positive pole, that is the anode, under the influence of an electric field. So what is happening over here? First we have put the DNA fragments over there spread on the gel plate, and it is now connected to the positive and the negative. So under the electric influence, since the DNA is negatively charged, it will start moving towards the positive end. Now the gel acts as a sieve and separates the DNA molecules based upon their size. That means the high density molecules will move slowly as compared to the low density molecules, that is the molecules with less base pairs will move faster as compared to the molecules with the more number of base pairs. Now this separated DNA fragments which are over there can only be observed, because DNA is the fragments which are obtained by the action of the restriction enzyme are comparatively very small. So first they have to be stained, and a stain which is used is ethium bromide. So the stain which is over here, which we are using over here, is ethium bromide. It gives a fluorescent color. So they become the DNA is stained with fluorescent dye, that is ethidium, sorry, it is ethidium bromide, and it is exposed to UV light, ultraviolet radiation. The strain, that is the DNA which is stained, appears as bright orange colored bands. So over here, if we just come to the overall, if we see, first the fragments of the DNA are cut with the use of restriction enzymes. Then they are laid on the gel plate. The gel is basically a matrix which is agarose matrix obtained from the seaweeds. This gel serves as a base on which the DNA fragments move, and it acts as a sieve also. Further, after laying it on the glass plate, the electric field is created, and since DNA molecules which are negatively charged will move from negative to the positive end, and because of the depending upon the size, and the size depends upon the number of base pairs. So the molecules or the fragments of DNA which are having high number of base pairs, that is high density, will move comparatively very slow as the fragments which are having less number of base pairs or the low density. But before we put them on the gel plate, the DNA molecules, the fragments of the DNA, have to be stained by ethidium bromide, which is a fluorescent dye. This fluorescent dye gives bright orange color when observed under ultraviolet radiation. So the UV light will give them bright orange color. will give them bright orange color, and the separated bands of DNA are now cut from the agarose gel and are extracted from the gel piece. So the next step is, after the fragments of DNA which have reached over there, the desired ones are extracted from this gel plate, and the process is known as elution. This process of removal of the purified DNA fragments from the gel plate after gel electrophoresis is known as elution, and the DNA fragments then obtained are used by joining them with the cloning vectors. So gel electrophoresis is a very important technique which is used for the purification of the DNA fragments, and now the DNA fragment is ready to be incorporated into the cloning vectors to obtain the rDNA.